Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Chris Woolsey from Business Review, and I'll be your host. It is our pleasure to have Namira with us today, who will be discussing Accelerate Your Time to Market in Nasal Spray Applications. Today's guest speakers are Pascal Farjas, Global Category Manager, Alain Regard, Technology Product Manager, and Celine Penticola, Customer Technical Support. I'd like to welcome you to our webinar platform on 24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. We will allocate some time at the end of the session to address any questions or thoughts that you may have. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. For now, please allow me to welcome Pascal. Over to you.
Thank you, Chris. Hello, everybody. We are very excited about this webinar as there are over 100 people attending. And among the attendees, we are happy to have people from the FDA and the NHS and other private companies and organizations from different backgrounds. I do really thank you for being here today and the interest you are showing. Let me first introduce Namira before entering in the topic of our webinar. For those who do not know Namira, we are a major player in the drug delivery device field. Our engineers and teams design and manufacture delivery systems dedicated to the pharma industry with a global footprint. The 770 million devices produced yearly or used by more than 100 million patients worldwide. So, what's differentiating Namira on the market today is our balanced business model, where customers can choose among our own IP products, or they can ask Namira to develop and industrialize their own IP products. And of course, we can also do a mix, as we are very flexible in our client approach. As you can see on the slide, Namira covers several administration routes. In fact, we offer multi-dose preservative-free multi-dose eyedroppers, various inhalers, insulin pens, and assistants for dermal applications, and of course, nasal sprays, which we will focus on today. And finally, we've developed off-the-shelf in vitro bioequivalence devices for some key nasal drug products. So, everybody, during this webinar, we'd like to take you on a journey for developing a device and, at the same time, present Namira's unique approach with regards to bioequivalence. We start by understanding the market and identifying the reference nasal sprays we want to target. Then, depending on each target, we isolate the most stringent regulatory requirements, which will then drive our in vitro bioequivalence program. From there, for each drug, we supply several batches of the reference from different markets, and we establish the identity of the device in terms of bill of materials, performance, patient use. After that, based on our pump platform, we develop a device with equivalent performance to the reference device with the reference drug formulation. And with that, we perform a comprehensive study comparing the test with a Namira device to the reference with either formulation and showing bioequivalence. Eventually, the whole study is gathered into what we call a data pack with a robust statistical analysis, which we provide to our clients. All the data generated can be used to support the drug registration process while justifying the device selection. And last but not least, we are ready to perform the same study with the final drug. So hopefully now you have a clearer understanding of the development journey for a bioequivalent device. So now we are going to taking that journey together. I shall be discussing market understanding. My colleague Céline shall be talking to you about regulatory, lab testing, and statistics. And Alain will be talking to you specifically on influence factors on spray characteristics. So in the following slides, I'm just going to take you step by step through that journey. Let's now start with market understanding. We focus our efforts on blockbuster drugs and take the patent expiry date into consideration. For each drug, we conduct specific market analysis to define our target in case several variants exist under reference. I will now show you the specific drugs we have focused on over the last few years. So, this slide shows our understanding of the market. Looking at topical nasal preparations as per the anatomical therapeutic classification, estimated at around $5 billion, you can realize at a glance that corticosteroids are the largest portion of the market. Let's remember that corticosteroids remain the first-line treatment for moderate to severe allergic rhinitis as the most effective option for relieving nasal symptoms. So, this market has been evolving over the last few years, relying mainly on two trends. We observed an increasing generic competition, particularly for some of the leading products within the market, like Nasonex. Then, more reference drugs made the switch from prescription only to OTC to extend brand life cycle and increase the addressable market while providing an accessible allergic rhinitis treatment for many patients who would otherwise go untreated. Sanofi's Nasacort AQ became the first intranasal corticosteroid in the US to switch to OTC in 2013. Overall, this mature market has experienced a limited but steady growth, and we expect the entry of new generics as well as improving access to healthcare will contribute to future market growth. That's why we've selected a number of corticosteroids as part of our bioequivalence program. I will now hand over to Céline, who will highlight the regulatory bioequivalence framework.
Thank you, Pascal, and hello everybody. In the first place, it is important to be aware of the complexity of the topic. As a matter of fact, different regulations are coexisting across the world. For instance, we will look at some strategic areas with different views of bioequivalence regulation. First, looking at USA, which is considered as the most stringent regulation. In other words, there are precise guidance per molecule, and when not, there is still a rigorous frame and global guideline to refer to. Our development work, previously mentioned and gathered into our data pack, is relying on US FDA guidelines. Then, let's go to Europe. In this case, there is also guidance. However, some points are not completely covered and open to interpretation according to different countries. In addition to that, the term generic is not always used in Europe, where the term hybrid is preferred. Now, I'd like us to focus on Brazil. Guidance is based on US FDA with some particularities, such as generic must have identical characteristics. The equivalence of US FDA in Brazil is called ANVISA. One challenge for a foreign company may be to find an agent who will be an intermediary when dealing with ANVISA. Finally, coming with China. China has similar philosophy to Brazil. For a foreign company with no office in China, they have also to pass through an agent to submit to the CFDA. There is no clear bioequivalence guideline for liquid dosage, and CFDA is trying to synchronize with US FDA, but guidance are still under construction. Things are moving quickly, but the approval can take a significant time, and it should still be challenging to find a local agent with the difficulty of the language. Now, I will say a few words on registration processes. First of all, whatever regulatory position of the targeted reference product is, prescription or over-the-counter but not under monograph, a generic product will follow the same process for its first submission, which means answer to the same requirements. Guidance could have different names, but contents are similar. When a client modifies his dossier, in other words, makes a variation of its first submission to add a second source, for example, there are two choices. The first one is to use the same process as was done for the first submission. Or the applicant can use a simple equivalence of the performances of the pumps without following the full guidance of bioequivalence for generics. This study could include only some criteria with no need to pass using statistical rules, but these criteria have to be inside client specification. For a generic company, it could be more relevant for authorities to compare their variation directly to the reference product. For example, imagine a door and you have a key for this door. Then we make a copy of this key for someone else. And we make a copy of this key. With this strategy, we will have less and less chance to open the door. This is why it is important to make only a copy with the master. To conclude, based on our experience, Namira could strongly support a submission dossier with the chapters linked to the device. Alain is now going to talk you through the third part of our journey, which is develop the design of the device.
Thank you very much, Céline, and hello, everybody. I will be talking about design of nasal sprays. As an introduction, I will start with some important drug and device considerations. The nasal spray products are the combination of two regulated components, the drug product on the one hand and the spray device on the other hand. The expertise and services offered by Namira are focused on the spray device. However, we have to take into account in our development the drug and device interactions. The device is a major contributor of the efficacy, patient safety, and robustness of the combination product. Over the next slide, I will touch on the criticality of the device performance and the work done at Namira to have it under control. Let's move now on the key function of a spray device. A spray device has three main functions. The first function is to preserve drug integrity. Leakage and weight loss testing ensure that the evaporation of solvent from the formulation is minimal, so that the drug potency remains constant over the shelf life. We also perform extractable profile studies of all materials of the device. These studies will feed in the toxicological assessment and leachable studies under the responsibility of the final applicant. In terms of design, the key element here is the selection of the raw materials. The second function is to deliver a defined dose of active ingredient. At the device level, this translates in delivering consistent shot weights of drug formulation through life of the product. Although this attribute is critical with regards to safety and efficacy, it is, in our point of view, quite easy to adjust and to control, and therefore will not discuss it further in the scope of this talk. The third function is to deliver drug product to the targeted site. Delivering the drug product to the nasal cavity in a consistent manner implies having consistent spray characteristics. Nasal sprays are characterized through, on the one hand, droplet size distribution, on the other hand, spray geometry, assessed via spray pattern and plume geometry. These attributes influence the deposition pattern in the nasal cavity, and as such, they play a role into the efficacy of the final combination product. In opposition, the percentage of small particles is related to the safety concern of the drug product entering the pulmonary tract. From all these functions we've been reviewing, the spray is by far the one which requires the most effort to control, and we'll now focus on the spray generation. Over the next slide, we'll review the impact of the pump, the spray nozzle, and the formulation on the spray characteristics. Let's start by the pump. The pump is the heart of the device. We discussed previously the measuring function of the pump. Another main function of the pump is to generate a flow with the flow rate and the pressure level allowing the generation of the spray. The flow rate and the pressure profile are dependent on the pump technology and the actuation profile. The nominal dose of the pump is also an influence factor as it impacts the spray duration. Here are some outputs of numerical modeling of one of our pumps. We model the pump with a dose of 100 microliter activated at 80 mm per second. The fluid used here is water, and the pump is associated with a given Namira spray nozzle. Part of the output of the study, we have here the flow rate and the pressure profile at the exit port of the pump. On the flow rate graph, we can see that the dose is dispensed in a less than 100 milliseconds, with the flow rate varying from 1 to 2 grams per second. On the pressure graph, we can see that this level of flow rate, in combination with the spray nozzle, generates an internal pressure raising up to 18 bars. This example shows that through modeling, we fully understand the physical behavior of the pump and that we are able to predict the outputs of the pump for given inputs. These outputs will be used as inputs for spray modeling I will see in the coming slides. So, moving from the heart of the device to the head of the device, we'll now focus on the tip of the nozzle spray, which is surprisingly complex. Let's see what's inside the nozzle tip. We have here a 3D view of the flow path in the nozzle tip. The liquid at the bottom here, driven by vertical channels, enter into the three convergent channels shown in dark blue. From there, the liquid dives tangentially into the swirl chamber shown in pink and will then exit through the spray orifice shown in green. I will be talking more about the swirl chamber principle in the next slides. The dimensions in this area are less than 1 millimeter, typically 0.2 to 0.4 millimeter. This image shows only a few dimensions. If we were to have them all, we'll end up with not less than 15 dimensions to describe the full geometry. These dimensions can be considered as 15 variables, which is a lot when considering this single feature. In this area, high precision manufacturing is required in order to generate a spray with a reduced variability at high manufacturing scale. Let's move now on the swirl chamber principle and the generation of the spray. We have here an interesting picture showing the velocity profile of the fluid. We have here a very good illustration of the swirl motion. In the swirl chamber, we can see the liquid rotating and converging to the spray orifice. The combination of the rotational and axial speed generates at the exit of the orifice a speed profile forming a rotating cone. The flow rate used for the simulation is 1.2 grams per second. This value is on the lower end of the flow rate for a pump actuated at 80 millimeters per second. As we can see on the flow rate graph I already commented. One thing to notice is the magnitude of the velocity at the exit of the orifice, 48 meters per second. This is the same as a car running at 170 kilometers an hour, or if you prefer, 105 miles an hour. And this value is nearly doubled when the flow rate reaches its maximum. At this stage, and just to wake everybody up, I have a quick question for you, and we'll see the answer on the next slide. So, the question is, what happened to the cone of fluid exiting the spray orifice at 170 kilometers an hour? Well, the answer is it breaks up into droplets in the first 2 millimeters after the exit. We have here two nice illustrations of the liquid breaking up and the formation of droplets. On the left picture, we can see in section an overlay of a CFD modeling result and a picture of a real spray. On the right picture, we have a 3D visualization of the fluid breaking up, once again from CFD modeling. So, now you understand the complexity of the atomization process and the importance of the swirl chamber. Let's move on to the effect of the fluid itself. The viscosity and the rheological behavior in a large manner of the drug formulation has a major influence on the spray characteristics and the surface tension to a second degree. As a matter of example, here are two graphs extracted from the literature. We have here 3 different spray devices tested with a mix of water and glycerin to cover a range of viscosity from 1 to 10 centipoise. We can see that the viscosity has a tremendous impact on the spray angle and on the droplet size distribution, represented here by the dV50 value. The batch-to-batch variability of the viscosity of the formulations can be significant and has to be taken into account when designing a spray to a defined target as required in the bioequivalent approach. When it comes to testing, you have to remember that the viscosity changes with temperature. The fluid memory effect has also to be borne in mind. As for example, shaking or not shaking the sample prior testing can impact the spray performance. You also might find that the delay between actuations can be an impacting factor. So, now you understand the complexity of the spray generation and how important are the pump, the spray nozzle, and the drug formulation to the spray performance. Finally, in my last slide, I'd like to give you some insight on the design tool and the methodology we are using in Namira for spray development. Over the last years, Namira has produced a tremendous effort in the nasal spray field in understanding the relationship between the input variable and the output performance. How are we doing that? What design tool are we using? Well, first of all, we are strongly relying on the experimental testing and the use of database to track the input and and performance data. Secondly, we can manufacture fully functional nozzle for testing purpose in a matter of a few weeks, including metrology control. This allows us to make design iteration loops in a short timing. This tool is also used to explore the design space. The next tool is fluid dynamics modeling. This tool allows us to understand the physics of the spray. We can use it to run some sensitivity analysis on specific parameters. I have shown you through this presentation several graphs and illustrations of some relevant outputs of numerical modeling. Last tool, but not the least, is mathematical modeling, making the link between the input variables and the output performance. Namira has a lot of experience in this area. We have successfully built mathematical models in the spray field on several projects. This approach, based on data analysis and mathematical modeling, allows an acute refinement of the design, with a final goal, to have the spray on target. Understanding the sensitivity of the performance and the design space allows the setup of an adequate control strategy. This to ensure robust and controlled performance in mass production. So that's it for the device design and spray expertise. Thank you very much for your attention. I will now hand over to my colleague Céline for the testing and method section.
Thank you, Alain. So, as Alain just mentioned, we will now cover the testing part of our journey. From the state of the art, Namira has developed its own laboratory of testing from cutting-edge technology. We developed our own testing methods to cover all the aspects of a nasal device. First, to know the performance of the pump. For example, test as dose accuracy, leakage, or weight loss. Secondly, to characterize the spray. Tests like, for example, droplet size distribution, spray pattern, and plume geometry. Then, we can control our production in terms of extractables. From these rigorous testing methods, we are able to either customize testing methods according to our clients' requirements or transfer them to a third party. I will now focus on the key spray performances for in vitro bioequivalence. These tests are very sensitive and use cutting-edge equipment which are automated and worldwide recognized. The first one is the droplet size distribution. As indicated in the name, the idea is to know the size of droplets into one spray. The second one is the spray pattern. It gives a top view of the shape of the spray and shows if the spray is homogeneous or not. The last one described here is the plume geometry, which also gives you a view of the spray but along its axis in order to estimate the angle of the spray. As I mentioned earlier, these testing are very sensitive, and we will see in the next slide that many parameters can influence the results. Indeed, here is a list, non-exhaustive, of areas where parameters could have an impact on the final results. As a consequence, it is important to understand the level of sensitivity of these parameters and to control them in order to maximize the chance to succeed in the in vitro bioequivalence. This work has given us sustainable and robust testing methods with a huge benefit in reducing the variability of measurements. For each field, I will give you a few examples to illustrate the huge range to be covered when developing a testing method. First topic is the environment. As equipment are based on laser, the enclosure system could have an influence if the signal is disturbed by the lights of the room. Secondly, maintenance of equipment. In other words, I mean, of course, calibration, but also regular cleaning of the lenses to avoid distortion of measurements. Thirdly, actuation of the device, which is probably the most important field. A key parameter is the actuation speed, or whether the device is holding into the actuator, or the way of priming the device, manually or automatically, are also significantly influenced. Then, if we are looking to sample preparation, for a nasal suspension, for example, shaking should have an effect on the measurement. We already discussed about actuation parameters, but the equipment themselves have also lots of settings parameters to be careful with. I will give you as an example the alignment between leather and nozzle. Last but not least, once the measurement is done, analysis of data is a huge part of the final result. In order to enhance the reproducibility and have reliable data, the measurement is done on the part of the spray that is fully developed, corresponding to the stabilization phase. You can see on the picture at the bottom of the slide the different steps of the spray from the formation through the stabilization phase to the dissipation. The duration of the stability phase is typically around 100 to 200 milliseconds. So once our data produced are reliable, let's move to the final step, the statistics. We will now focus on main differences in statistical approach. Our intent is to illustrate the main differences, again to enhance chance of success by knowing better the requirements and not to take position. On one hand, there is USA and countries that follow FDA guidelines, which are using PBE calculation, which means population bioequivalence. In this case, the calculation is complex, but everything is detailed in a formal frame. On the other hand is Europe. They will strongly recommend to use ABE calculation, which means average bioequivalence, but no guidance are available on a statistical point of view. So PBE, ABE, what does that mean and what are the differences? In the first place, PBE and ABE are not looking at the same thing. Let's try some simple introduction of PBE, ABE theories. For PBE first, from formula, we have a comparison of means correlated by the variability of the reference device. From testing, we have access to mean and standard deviation of a part of the population, whereas we need to extrapolate to the whole population. That means building an interval of confidence. However, this is something which is not possible on a ratio. This is why we have to linearize the formula. This leads to have an interval of confidence strictly less than 0. So, as the lower bound of the interval of confidence will always be lower than the upper bound, we will only have a look on the upper bound of the interval of confidence. Then for ABE now. The principle is much more simple. The aim is to calculate an interval of confidence of the means difference between test and reference. And the two bounds of this interval of confidence have to be inside bioequivalence limits plus or minus theta. Let's move to some concrete examples. So calculation, theory, okay, but what does it mean concretely? The example on the left side are cases where ABE calculation is accepted. Case number 1, some variability between test and reference and a certain gap between the means which is acceptable to pass the bioequivalence. Case number 2, when there is an heterogeneity of dispersion between test and reference, the maximal delta between the means becomes much lower. On the right side, the example will be where ABE calculation is rejected. Case number 1, in this case, the delta between the means is slightly higher than the previous case. The last case is atypical, as means are very well aligned, but it fails. This is because the variability of reference is so huge that the interval of confidence overtakes the limit. What about PBE? So what we observed for PBE is that the higher the variability on the reference product, the higher the probability to pass bioequivalence, and especially when the variability on test product is low. The two examples on the left are illustrating this point. There is one counter example on the right side where PBE is rejected, but you can observe that the variability of the reference is not so big compared to the left side. The next slide will give you some clues for understanding the main differences between ABE and PBE. This graph illustrates the series of ABE and PBE. The x-axis represents the relative standard deviation of the reference. The y-axis represents the maximum delta acceptable to pass the bioequivalence. In other words, it corresponds to the maximum ratio between means of test and reference. For ABE, as we are only comparing means, whatever the relative standard deviation are, the maximum delta acceptable is a straight line. The usual limit is 15%. For PBE, the results are strongly dependent of the variability of the results and much more from reference. I will take two examples using the same variability for the test fixed at 10%, which means the orange curve. The first example, with the variability of the reference at 5%, gives a delta maximum for the means of 10%, which is lower than ABE, which is at 15%. In this case, PBE is less permissive than ABE. The second one, with a variability for the reference at 20%, gives a delta max for the means around 28%, which is much higher than ABE, which is still at 15%. In this case, the PBE is more permissive than ABE. To conclude this statistical chapter, Namira has developed statistical tools to help into the laboratory results interpretation and to understand where could be the area of improvement to enhance chance to succeed in vitro bioequivalence. Pascal is now going to walk us through the last part of the webinar.
Thank you, Céline. So, everybody, to conclude, the journey to developing a device with in vitro bioequivalence performance is more difficult than it seems. We have shown you how we, in Namira, can answer the complexities of the nasal equivalence requirements. How are we doing that? Firstly, by understanding multiple regulatory requirements. As we've seen before, the regulatory framework is very complex, and the requirements vary from one region to another. Whatever the pharma company regulatory strategy is, either for launching a generic or dual sourcing, prescription or OTC, Namira can support the registration process on the device side. Secondly, by developing devices with cutting-edge design tools on spray science. Because the efficacy of the drug really depends on the device's ability to deliver a uniform dose as well as a reproducible droplet size and plume, the drug delivery device is a critical element for nasal spray performance. Namira brings into play its expertise, in particular regarding spray customization, which is surprisingly complex, as you could see, not only on the design side, but also in production, where high precision manufacturing is required to achieve a reduced spray variability. My colleague Alain also discussed how the drug properties can directly impact on the spray characteristics with its own batch-to-batch variability and has to be taken into account within the development of the device. Thirdly, by setting up customized testing methodologies with state-of-the-art lab equipment. We acknowledge that with different testing methods, we have different test results. Any lab testing methodology has to be considered for each device technology. In other words, one testing method for a given device will probably have to be adapted for another device. Céline also discussed that multiple different parameters play a role in the test results, such as the actuation profile during the test or the analysis of the generated data after the tests. That's why Namira invites the clients to discuss about lab tests and share methodologies as from the very beginning of a project, so that we have a greater chance to pass bioequivalence. Last but not least, by mastering statistical requirements and models with internal experts. Céline showed the main differences in terms of statistical approach per region. The FDA looking at PBE with a quite complex calculation, whereas the NEA rather looking at ADE. Those two calculation modes are quite different. What's important to remember here is that Namira well understands both statistical approaches and can support clients whatever the calculation mode they follow. So, Namira is a partner to work with, and our clients recognize the keys to success, or cooperative relationship, commitment, technical competencies, and responsiveness. Through our customers, we really put patients first. And to illustrate that, Céline is the main technical contact for our clients, responding to all their inquiries on any bioequivalence project. I will now hand over to Chris for driving the Q&A session.
Thank you very much. Just a reminder for the audience, in order to ask questions, you can send them in via the questions widget just type them into the box at the top left-hand corner of your screen and click Submit. I see that a few questions have already come in, so go straight ahead. And the first one will be, what's in the data pack? So what is in the data pack? Who would like to answer that one?
Yeah, I can take this one, Pascal speaking. So the data pack is a very internal word from Namira. This is a compilation of the in vitro bioequivalence study comparing the test to the reference. You will find the characterization of the reference drug, for example, the bill of materials, the performance, design, but also instructions for use, as well as the characterization of the Namira device, obviously matching the required performance. All that has been done with the drug formulation of the reference. And then there is a comparison between different regulations across the world and why we selected the FDA guidances in this document. Also, the test results are detailed with a statistical analysis when required, as well as all the testing methodologies that Namira used to achieve those results. So basically, there is one data pack per identified drug, and we share this document under a confidentiality agreement with our clients.
Thank you. Okay, I see another one here. How do you make sure your manufacturing process is so precise, in particular for the swirl chamber dimensions? Who would like to answer that one?
I will be taking this one. Alain speaking. Well, I understand the question. There are two folds. The first fold is the precision of the manufacturing, and secondly, the measurement aspect. Regarding the manufacturing aspect, there are a number of prerequisites to be met in order to achieve a precise manufacturing. So one of them is the toolmaker selection. There are only a few toolmakers that can make this kind of tools with such high precision and with the minimal intercavity variability. Secondly is the process development that we are doing during the mold qualification through design of experiments on injection settings. So this is really the heart of the experience and the experience of Namira. And then also the process control that we are putting in place into production afterwards. The second point is the actual measurement itself. We are using mainly two different tools. Firstly, Namira is equipped with a Metrotome equipment which provides high precision CT scan. With this tool, we can, on assembly of multiple components, take measurement and detect surface defect. And secondly, for each component, we are using optical and contact measuring machine. In this field, we have developed our own methods in order to achieve the level of repeatability, reproducibility required for the spray nozzles. So this is all part of our expertise.
Thank you for that. What is the timeline to develop a bioequivalent device? Sorry. So once again, what is the timeline to develop a bioequivalent device? Who would like to answer that one?
I will be taking this question again. Alain speaking again. As already said, Namira has already developed off-shelf devices for key identified drugs. For other drugs, starting from scratch, we have developed our own tool to reduce the number of design loops in order to get sample ready for preliminary testing below a year time. And as it stands, for the typical lead time to have sample ready for registrations, this means that they can be also used for stability and bioequivalence testing, is 12 to 18 months. So this is the typical lead time we are running on today.
Okay, thank you. I see another one here. When the statistical calculation is not okay, can Namira help in achieving in vitro bioequivalence? Who would like to answer that one for us?
I will take this one, Céline speaking. So based on our work done to understand what is behind the statistics, we have developed statistical tools to help us in the interpretation of the results. So in other words, from our statistical analysis, we are able to guide our clients on where could be the area of improvements to succeed in the in vitro bioequivalence. We will strongly support our customers, as I previously said, in the way of understanding the part of the statistics of their dossier.
Thank you. I see another question here. What controls do you have in place for routine batch release? Again, what controls do you have in place for routine batch release? Who would like to answer that one?
Alain speaking. So beyond the component and subassembly dimensions we are controlling, for the pump we are controlling the priming and the dose consistency. And for the nozzle, we are checking the droplet size distribution and the spray pattern. We can also supply extractable routine batch release analysis upon customer request. For that, we have developed the analytical methods, and we have all the equipment to provide this service on demand.
Thank you. I see a question here. How is Namira able to perform testing with customer formulations, and how do you do that?
So I will answer, Céline speaking. So what I can say that our laboratories have a dedicated area for testing the active ingredients. So first, we have to perform some pre-testing to confirm the testing methods. So either a customer or a Namira method, if it is well adapted to our device combined with customer formulation. Once these verifications are done, we are able to perform the testing on pumps. So it means shot weight, priming, repriming, and on some spray performances like DSD, droplet size distribution, spray pattern, and plume geometry.
Okay, thank you for that. And that will bring us to the end of the Q&A session. Apologies if we didn't get time to answer all your questions. One of the speakers will follow up in their own time in the future. So that just leaves me to thank Pascal, Alain, and Céline for what was a great presentation, and to thank Namira for sponsoring this webinar. To the attendees, you'll receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned, and follow us on Twitter at @BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. Thank you once again, and I hope you all have a nice day.